Hi, my name is Alexa Schmitz, and I'm a postdoc with the Barstow Lab in the Department of Biological and Environmental Engineering at Cornell University. Many thanks to the organizers for the invitation to present my work here. Generally, the research in the Barstow Lab aims to use specialized microorganisms to solve problems in renewable energy. My work in particular is focused on engineering microorganisms to recover rare earth metals, which are often critical ingredients for sustainable energy infrastructure. There are 17 rare earth elements, which include the 15 lanthanides, plus scandium and yttrium. On the right here are 5 of the most critical rare earth elements: neodymium, dysprosium, europium, terbium, and yttrium. Rare earth elements have excellent electronic, optic, catalytic, and magnetic properties that provide solutions for many challenges of modern technology, making them useful for a wide range of applications from cordless batteries and power tools to smartphone displays. Of particular note, rare earth elements play an important role in many emerging clean technologies, such as energy-efficient lighting, rechargeable batteries, electric cars, and especially wind turbines, where they are used as strong, lightweight magnets. So as the world moves towards a global renewable energy system, popularity of hybrid and electric cars, wind turbines, and compact fluorescent lamps has led to an increase in the price of and demand for rare earth elements. Thus, those of the most importance for clean energy have also become those with the greatest supply risk. In 2011, the Department of Energy has declared 5 rare earth elements as critical minerals due to their importance to clean energy. But as of 2018, the DOE has now declared all rare earth elements as critical minerals. Despite their name, rare earth elements are not actually rare, just very dispersed throughout the Earth's crust and thus difficult to mine directly. A major hurdle for increasing rare earth production is the embedded cost, both economically and environmentally, for extracting the minerals. Traditional physical chemical extraction methods require very high temperatures and harsh chemicals and produce a large amount of toxic waste. So as a result, environmental regulations and remediation costs have led to the closing of major mining companies in the US. But as a promising solution, biological approaches could supplement or replace the traditional physical chemical rare earth extraction methods with much more environmentally friendly methods that can be done at ambient temperature and utilize biodegradable and aqueous chemicals. Such a replacement would greatly lower the carbon footprint of the renewable energy technologies that are relying on rare earth metals, especially electric vehicles and wind turbines. But bioleaching of metals is not new. It's actually used currently to extract 5% of the world's gold and 20% of the world's copper. Pictured here is the Escondida mine in Chile, where 1.2 trillion liters of low-grade ore is heap leached by iron oxidizing bacteria to produce soluble copper. Bioleaching of rare earth metals is done with organic acid-producing bacteria and fungi, including Aspergillus, Pseudomonas, Gluconobacter, and can be used to extract rare earth elements from primary sources, such as monazite ore, or from waste and end-of-life materials, such as coal fly ash and fluid catalytic cracking catalyst. So one of the most efficient demonstrations of rare earth bioleaching was done by a team of researchers at the Idaho National Lab who used the bacterium Gluconobacter oxydans to bioleach rare earth metals from spent fluid catalytic cracking catalysts from petroleum refineries, as well as retorted phosphor powders from fluorescent bulbs. Gluconobacter oxydans is a gram-negative bacterial species, ubiquitous on flowers and fruits, non-pathogenic. It's actually classified as generally regarded as safe for use in foods such as kombucha. All of this, along with its ability to tolerate low pH and that it can be genetically modified, make it an excellent candidate for engineering an efficient rare earths bioleaching system. However, its efficiency as it stands now is still too low for commercial application. The primary mechanism by which Gluconobacter solubilizes rare earths is to greatly lower the pH through the production of biodegradable organic acids, such as gluconic acid from glucose. However, experimental results have indicated that other yet unknown factors may be playing a significant role as well. So the goal of our lab's research on rare earth extraction is to improve the overall bioleaching mechanisms of Gluconobacter by targeting the genes that underlie production of gluconic acid, but also any of these other important factors that we might be able to identify. So targeted genomic improvement requires a priori knowledge of the genomic sequences underlying a trait of interest, such as rare earth bioleaching. Despite extremely advanced tools for reading and writing genomic sequences, the function of over a third of the genome sequences today is still unknown, which leaves us asking which genes to target to improve the efficiency of Gluconobacter's rare earth bioleaching. The best way to determine if a gene is important for a trait of interest is to disrupt its function, then test if the trait is affected. To do this for rare earth bioleaching, we have created a single gene knockout mutant for every non-essential gene in the Gluconobacter genome. This is called a whole genome knockout collection. And we did this using a powerful and low-cost process developed in the Barstow lab called Knockout Sudoku. So to start, we created a plasmid with a mutating piece of DNA called a transposon, which also contains a gene for resistance to the antibiotic kanamycin. We inserted it into E. coli for safekeeping and replication. We then grew the E. coli containing the plasmid and the Gluconobacter overnight. Then we mix them together on a petri dish where they mate, which allows E. coli to transfer the plasmid to Gluconobacter. The key here is that Gluconobacter can't replicate the plasmid, and since the transposon has a kanamycin resistance gene, the only way it can survive on kanamycin is if it successfully inserts the transposon into its genome. Thus, we can select for genomic insertion mutants by challenging the Gluconobacter with kanamycin. For this particular transposon, hundreds of thousands of different insertions are possible. Some may go into genes, while others may go into regulatory sequences, or sequences in between genes. And some will actually go into the same gene more than once, but often in a different location. Gluconobacter oxydans has around 3,200 genes, but clearly picking just 3,200 mutants would not mean I've hit every gene. So using a computer algorithm that runs a Monte Carlo simulation of the genome insertions into Gluconobacter oxydans genome sequence, I estimated that I would need to pick about 50,000 mutants to recover at least one in each non-essential gene in the genome. Ordinarily, picking 50,000 bacterial mutants is a daunting task. With the help of this robot, I could pick all of them in only a few days. The robot locates the colonies through image analysis and uses metal plates to transfer them to liquid media on a 96-well microscope, as you can see in the back of this video. In total, I picked 524 microplates of mutants, although pictured here is only half of them. I then used barcoding PCR, combinatorial pooling that resembles the game Sudoku, next-gen sequencing, and a Bayesian location inference program to identify what mutant is located in each well of the 524 microplates. Finally, after a ton of work with much help from the rest of the lab, I selected 1 to 2 mutants per non-essential gene to curate a whole genome knockout collection for Gluconobacter oxydans. Overall, I was able to identify at least 1 mutant for 2,733 genes out of 3,283. The remaining 550 non-mutated genes are likely essential genes whose disruption would preclude bacterial growth and recovery. So once I had the whole genome knockout collection, I could test the mutants for any that disrupt or otherwise modify rare earth bioleaching. pH has been shown to be a good indicator of bioleaching capability for Gluconobacter. Thus, I can monitor acidification of the media to identify potentially important mutants. To do so, I have developed an assay using the pH indicator bromophenol blue, which begins to change color from purple to yellow around a pH of 4.6. Shown here, a test tube with 2% glucose and bromophenol blue will change completely to yellow with the addition of Gluconobacter and overnight incubation. Using the bromophenol blue assay, I have tested all 2733 gene knockout mutants for their rate of media acidification. To do the assay in high throughput, I first mixed the solution of water, glucose, and bromophenol blue in a 96-well plate, then added to that a small amount of bacterial culture from a collection plate. Doing so caused the pH to immediately begin dropping, and the bromophenol blue would begin changing color to yellow. I could use a plate reader to measure the absorbance of 600 nanometers light by bromophenol blue over the course of about 10 minutes. I then used the program for the plate reader to calculate the rate of change in absorbance. Then, considering that the majority of bacterial mutants on each plate should be unchanged in their ability to modify pH, I identified important mutants by looking for outliers in their rate of acidification. So for example, in this plate 130, wells A4 and B4 contain the same mutant strain, which decreased the pH at a significantly faster rate than the majority of the plate. As you can see by the color, after about 1 hour here, the bromophenol blue in those wells turns yellow sooner than the others. As another example, on this plate 162, 6 wells contain the same mutant that was actually slower at decreasing the pH. In fact, the bromophenol blue in these wells, despite normal bacterial density, actually never turned to yellow, stayed blue. So using the bromophenol blue screen, I identified 74 gene knockout mutants with a significantly different rate of acidification. Those represent basically a putative comprehensive set of genes underlying acidification. So 30 of these mutants were faster at dropping the pH, representing genes that would normally attenuate acidification. 43 of the mutants had a slower rate of pH drop, and these represent genes whose function would promote acidification. I went on to verify these results with greater repetition and a direct comparison with a pseudo wild type. I found of the 74 mutants identified through the large scale screen, 29 still had a significantly different rate of acidification, 19 of which were faster and 10 of which were slower. In fact, 2 in particular had exceptionally faster rates of acidification, almost 5 times faster, indicating that knockout of the genes in these mutants may already result in considerably improved rare earth bioleaching. So in addition to measuring rate of pH drop, I would also like to measure endpoint pH using the thymol blue pH indicator. While faster acidifiers would improve efficiency as they would require less inputs, mutants that can lower the pH even further than wild type may produce a biolexiviant with overall improved bioleaching capabilities. However, it's still uncertain if the final pH is really limited by the acidification mechanisms genetically or actually by the tolerance of the bacteria to low pH. But finally, once I've identified the suite of mutants affected in media acidification, I will use rare earth containing waste materials. In particular, I have retorted phosphors from old lamps and ICP-MS to conclude which mutants are truly changed in their ability to solubilize rare earth elements. Once we've identified genes underlying rare earth bioleaching, we will use directed evolution to target their regulatory regions for improvement. So following from the example that I gave from the plate, if knocking out gene A reduces acidification and rare earth bioleaching, we would target the regulatory region of gene A to increase its expression. Conversely, since getting rid of gene B enhanced bioleaching, we would target its regulatory regions to decrease expression. Ultimately, through the up and down regulation of multiple important genes at once, we hope to engineer a new and improved Gluconobacter oxydans with a high enough bioleaching efficiency that would allow for its widespread commercial application in the environmentally sustainable recovery of rare earth elements. This work has had essential contributions from many mentors, colleagues, collaborators, and funding sources, for which I am immensely grateful and could not have done without. Thanks again to the organizers for inviting me, and thank you for taking the time to listen to my presentation. I really very much look forward to hearing about everyone else's work soon.